It's not only news, it's science breakthroughs. Tomorrow's technologies. Advanced medical techniques. It's a glimpse into the future where we discuss artificial intelligence, earth and energy, enhanced humans, future society, health and medicine, space, robots and machines. Welcome to Science Bytes with Joe and Craig.
Welcome to Science Bytes. Today is Thursday, April 23rd, 2020. At the top of our show today, we are going to discuss the use of gene drive that may disarm a deadly wheat pathogen.
A gene drive is a genetic engineering technology that propagates a particular suite of genes throughout a population. Gene drives have been proposed to provide an effective means of genetically modifying specific populations and entire species. Joe and I have discussed this on our radio show, the Joe and Craig Music Show on WGOT, in the past.
That's the scary part. If this concept is weaponized, there are benefits from this technology like helping plants.
The Fusarium fungus is the misery of every wheat farmer. The fungus causes wheat scab, also known as head blight. It decimates harvests and contaminates grains with a toxin harmful to people and animals. Now, Australian researchers have come up with a new strategy to combat Fusarium graninarium, the most notorious wheat scab pathogen. In the lab, they have used a gene drive to get rid of the fungal genes that make this pest so toxic.
I'm not going to say that word.
I don't blame you.
Fusarium graninarium. Oh, whatever. Okay. The new wheat strategy would be the world's first use of the gene drive to control a pathogen in plants. The findings are very enticing for both plant and human health. To date, gene drives have never been deployed outside of the lab.
Wheat scab is a growing problem in North America, Europe, and China. Researchers are scrambling to breed wheat resistant to this fungus, with some recent success. Even so, disease management is reaching a crossroads. It takes a lot of time and effort to develop new breeds of wheat, and producing significant resistance to this fungus will likely require introducing multiple genes. Even then, complete protection may not be achieved. Meanwhile, the fungus rapidly becomes resistant to any chemical treatments, and various countries are beginning to ban the use of fungicides. For those reasons, researchers say it's important that we don't shy away from considering new and novel methods for managing diseases.
The gene drive process involves introducing DNA into the organism that causes one version of the gene to be passed to the next generation, but not to the third. Eventually, just the desired version of those genes remain in our population.
It's really fascinating that they can do that.
I know.
Scientists typically use gene editing tool CRISPR as the gene driver. That's how researchers hope to fight malaria. They adapted CRISPR to spread a gene that transformed populations of a malaria-transmitting mosquito into all males so the species cannot reproduce. Given the many uncertainties about the long-term consequences of releasing a gene drive, scientists are proceeding cautiously with such work. Although aware of those concerns, researchers still felt a gene drive was worth exploring for wheat scab. Their intent was to get rid of the 3 Fusarium genes that make the pathogen highly infectious and the infected grains toxic, while all leaving the fungus otherwise intact, at least DNA-wise.
They found that CRISPR did not effectively spread the harmless version of these genes. A gene in another fungus did, and made it easier for us to now work with it.
Yeah, I can definitely see this being weaponized really badly.
Oh yeah, be careful.
The researchers linked that gene to safe versions of the 3 targeted genes. Once in the Fusarium, the gene drive gene caused any sexually produced spores that wound up with the original versions of the targeted genes to die. Thus, the innocuous versions were transferred to the next generation. Those subsequent generations were less able to cause the wheat scab, but otherwise were no different from the typical Fusarium.
In just 3 generations, the 3 toxic genes were completely gone. We're going to keep our eye on this as it develops.
Yeah, it's going to be really interesting to see where this goes.
Yeah, I'm curious on this one. Well, we're going to take a short break, and when we return, we will discuss a new finding regarding Alzheimer's. As we discussed before the break, a gene variant may drive back Alzheimer's in some people.
People with a gene variant that puts them at high risk for Alzheimer's disease are protected from its devastating effects if they also carry a variant of a completely different gene. Stanford University School of Medicine investigators reported in a large new study.
The findings suggest that a substantial fraction of the estimate 15% of Americans carrying the high-risk gene variant are protected to some degree from Alzheimer's disease by the variant of another gene.
A gene will often come in a variety of versions or variants that can produce different traits. The findings also may help drug developers better identify clinical trial participants and treatment for what remains a disease without a cure, despite billions of dollars spent in the quest for effective therapies. About 5 million Americans, including roughly 1 in 10 people age 65, have symptomatic Alzheimer's disease. Even larger numbers have a precursor called mild cognitive impairment. About half with this condition move on to full-blown Alzheimer's. There are medications that can slow the development of cognitive symptoms somewhat, but no available drugs prevent the disease's progression or extend patients' lives.
What causes Alzheimer's is not totally understood. There are probably numerous factors. Scientists have known for 3 decades about one main contributor to the disorder, a gene variant, APOE4. That's more than 3 times as frequent in the Alzheimer's patient than among people without the disease.
A hallmark of Alzheimer's is the aggregation in the brain of gummy deposits, or plaques, composed of a protein called beta amyloid. Amyloid aggregation starts more than 10 years before symptoms appear. Recent technological advances have enabled the early prediction of Alzheimer's onset by analyzing beta amyloid levels and other protein levels in the cerebrospinal fluid, and by detecting the buildup of Alzheimer's plaques in the brain via imaging. These biomarkers make it possible to predict the disorder's onset before outward symptoms become apparent. Yet, even having 2 copies of APOE4 by no means ensures that a person will develop Alzheimer's. Some such people live to an age of 85 or 90 without any symptoms. They're protected somehow from the debilitating effects of this gene variant.
Researchers have focused on a variant of a gene for a protein called klotho. High blood levels of klotho predict longevity in animal studies. There is also evidence for this in humans.
Learning more about how the protective gene variant works may also lead to a more sophisticated understanding of APOE4's debilitating effect on cognition and, importantly, help researchers to zero in on therapeutic targets for the prevention or mitigation of those effects.
Now we're going to move on to those things that Craig and I just can't stop thinking about this week. Craig and I keep talking about this mutant enzyme that can break down plastic bottles in just hours.
This is really cool. The enzyme can break down PET plastic bottles into their individual chemical composites, which could later be reused to make brand new bottles. Conventional recycled plastic goes through a thermomechanical process that isn't high enough in quality and is mostly used for other products such as clothing and carpets. So this enzyme can break down 90% of PET polymers in just 10 hours. The new enzyme was first spotted in a heap of composted leaves back in 2012.
Researchers are hoping to test the materials, both industrial and commercial, in France in 2021. I guess we can't let this go because we keep wondering if this concept can be expanded to take care of all our garbage us humans continue to make.
I don't know, Joe. Can you imagine if they can just get this and just spread it around a landfill and just let it eat all the garbage or whatever it does?
It's going to be amazing. Bring it back to, I think you want to say, molecular.
Yeah, that's it. Molecular. To bring it down to its molecular structures. That would be really, really cool.
Sure would. Okay, that brings us to the close of another episode. So thanks for listening to Science Bytes with Joe and Craig. We will be back again next week with a new episode. You can find all our episodes of Science Bytes for free on all podcast platforms by searching Science Bytes with Joe and Greg.
Joe and I are grateful to you, our listeners. Let us know how we're doing by leaving a review and giving our show a 5-star rating. It really helps to move up our rating so more people can find us. Thanks again for listening, and we'll see you next week.